Hello everyone. It's a pleasure to be standing in front of you and talking about like our research. And during the talk, I might refer like I did it, but always remember it's not just me doing it. It's a team effort. All of us had like worked on this project for a couple of years to get this revolutionary idea out. So I'll today talk about monocot grafting, which is a very novel and emerging tool that can future-proof crops for changing climate and make crops more climate resilient. And during the talk, I'll talk about what exactly is grafting and talk about the aspects of grafting, that it is ancient, but it is also used in agriculture these days. Then I'll talk about like a recent work on grafting and how it has overturned the long-standing consensus that monocotyledonous plants cannot be grafted. And then I'll talk a little bit about where we are taking things now and in which future direction we are using monocot grafting and how we are using it to answer some of the important biological questions. So first thing first, grafting is the horticultural practice by which you fuse two plants so that they can grow as one. And you might have seen like a gardener where you have like stem cutting of a rose or one plant and add it to the rootstock of another, and that grows together as one plant. And there is evidence of grafting or like some of the scripts in China, in Greece, and then in Rome. And there's also mention of grafting in Bible and the Book of Mormon. So it's really ancient, and there's been reference to grafting all along through ages. And if we think about like the economical benefits of grafting, in modern time, about 2 billion plants are grafted globally per year, mostly like at the level of orchard plants, like whether you talk about apple or you talk about some pears and grapes. And let's look at a few examples. So these are trees in Gloucestershire forest. And what you can see is there's a birch tree and a beech tree that can actually like inosculation, which is a Latin word for like kissing inwards, but the word for the day is grafting, so don't worry about inosculation. Worry more about like grafting. And there's another like example of grafting where a host plant is actually grafted at the root shoot junction by a parasitic plant striga. So we're looking at this parasitic plant striga, and you can see them as like pink flowers here in the field of sorghum and maize. And what this parasitic plant does is it forms grafts with the vasculature of the host plant to just draw up nutrition. So this is a mechanism for the parasitic plant to draw all the nutrition from xylem and phloem. These are these transporting units in plant, and get all the nutrition. In more recent time, if you think about the grape wines or where we get the wine from, that is also an excellent example of grafting, because in the 19th century, the grape wines were being devastated by this insect pathogen called phylloxera. And back in those days, the rootstock of a resistant grape was added to the juicy grapes that we were getting from the grape wine, and that provided resistance to the grape wine that's present to date. So let's think about monocots and dicots now, and think a little bit about the anatomy, what enabled grafting. So dicotyledonous plants are plants that have two seeds, and if we cut a cross section through the stem, we can see this arrangement of vascular bundles, and then there's this special tissue called vascular cambium that's present in the stem of dicotyledonous plants. And if you look about the stem sections of monocotyledonous plants, you can see that these vascular bundles are not arranged, but they are like scattered all throughout the stem, and there's no vascular cambium in monocotyledonous plants. So there were two reasons which were thought was enabling grafting. One, this nice arrangement of vascular bundles in dicots, and also the presence of this vascular cambium, which was not present in monocots. So it was thought that monocots cannot be grafted, and there was like reports from back in 1913 all the way till 2020 saying that it was impossible to graft monocots. And we were a bunch of like weird little lab rats thinking about, really, monocots cannot be grafted? What do these scientists know? Let's put it on Google. Can monocots be grafted? And the first question we put on Google, can monocots be grafted? And it says, within just a fraction of a second, of course, monocots cannot be grafted. But I'll give you a spoiler alert. We've developed a protocol where we can graft monocots, and that's why I'm standing here today and talking about why it is an emerging tool and how it can answer important biological questions. And if you want to be a little more experimental with Google, you can actually type the question again and ask, can monocots be grafted? And the first thing it says is, yes, it can actually be grafted, and it refers to our paper. So woohoo, Google has actually corrected the facts. So now let's talk about the different method that we thought of grafting monocots. So we tried a couple of methods, and it was probably a serendipity. And we looked at the embryonic tissue of the monocotyledonous leaf. And when we look at the embryo, there's this embryonic shoot and the embryonic root. And instead of grafting the stem that was done in dicots, we actually started grafting the embryonic tissue between the root and the shoot. And when we grafted the embryonic shoot to the embryonic root, the plant actually grew into a grafted plant, and there was functional connections between the xylem and phloem. So the plant was actually grown to be a functional plant with these successful connections in xylem and phloem. So let's look at a schematic view of how we were grafting the seed. So we were literally like plant doctors with these biopsy punchers. We looked at the seed, went at an angle to the embryo, and we did like this precise incision between the root and the shoot junction, and replaced the shoot part of the seed with the shoot embryonic shoot part of another seed. And if you look at an example of a grafted plant, so this is a rice that's grafted, and this is about 2 months after grafting. And what you can see is that there is this graft junction shown by the white arrow, and these vascular connections, or like this vascular cylinder, was established after graft junction formation. So we wanted to test whether these grafted plants were actually functional or not. And how we went about doing that was using a fluorescent dye. So there's this fluorescent dye called CFDA. And we wanted to establish whether the phloem was connected or functional or not. So for that, we applied 1 drop of this fluorescent dye on the tip of the shoot, and after about 30 minutes, we were able to cut sections at the root and see whether we did get any fluorescence in the root. So if you're getting any fluorescence in the root, it's because of transport of this fluorescent dye from the shoot to the root. And what you can see that both in grafted plant as well as in non-grafted control, you can see that the fluorescence is seen in the root. And when we did sections of this, so there's fluorescence in the phloem region of the non-grafted plant as well as grafted plant, so which establishes that these plants have vascular connections and they are actually grafted. So next we wanted to see whether the xylem was functioning and could things be transported from the root to the shoot, and we did the reverse. So this time we added the fluorescent dye to the root tip and did sections of the shoot tip after about 30 minutes. And what we were able to establish in a similar way was that we were able to get like a fluorescence both in the non-grafted as well as grafted control in the shoot. And if we did a cross section, we can see this fluorescence in the shoot, in the xylem elements in both the grafted and the non-grafted control. So it was quite nice that we were able to like establish this protocol of grafting plants. But we were quite interested to understand like what were the molecular mechanisms that were driving this grafting mechanism. And for that, we actually looked at this region, so specifically where we were grafting the plants and growing them from 1 to 7 days, and taking sections and doing RNA isolation and deep sequencing to understand what were the genes that were upregulated in a grafted plant compared to a non-grafted or a wounded control. And I won't go into details, but just tell you the point that we did some gene expression analysis and hierarchical clustering of these differentially expressed genes. And what we actually came to the conclusion was that there was a lot of like cell division and cell wall remodeling happening in the initial stages of this graft union formation, followed by changes in some of the auxin and gibberellic acid responses, causing this functional graft formation, which was also followed by xylem and phloem differentiation and active elements of xylem and phloem forming at the graft junction. So then we wanted to be a little more curious because we did that grafting in rice, and we were like, can we graft other species? So we started with wheat, and here we are looking at 2 different wheat types. So one is a wild type, which actually does not produce like this blue color. And then the other is a transgenic wheat, which produces a Gus reporter, and that's why you see this blue coloration. So we grafted the GUS reporter wheat, which is expressing that GUS gene with the wild type wheat as a root. And we also grafted that as a rootstock and the scion. And what you can see here is that we were able to get like these successful grafts with the vascular junctions being functional, and similar to establish the vascular connection assay, we applied CFDA both to the shoot and to the root, and then assessed whether the fluorescence was present in the opposite tissue type or not. So we were able to establish grafting in wheat. So then we were quite happy because we were able to establish grafting not in rice but also in wheat. So next we wanted to be a little more exploratory and look at the whole phylogenetic tree of the monocots. So if you think about monocots, there are these three distinct clades, the elasmatids, the lilioids, and comelids. And what you can see in these different colors are the 11 orders of across the whole monocotyledonous clade. And what we did then was looking for raiding all the stock centers of seeds, going to Scottsdales a couple of times on the weekends, looking on Amazon everywhere to get the seeds for all the different species that we could acquire across this whole monocotyledonous clade. And this is just an example of like the different species that we grafted. So we grafted species from poils, gingerbils. I won't go into like all the details, but you can see that we grafted like 78 seeds with almost like 70% success for wheat. And then you have like some of the seeds that are like for Zea maize, like rice, and then also the different examples are here. So what was more prominent for us was that we were able to like not even graft rice and wheat, but we were able to like graft tequila agave, banana, date palm, oil palm. So we were actually able to graft 9 out of those 11 orders of monocots. And we were able to graft 9 out of the 11 orders because I think we didn't get seeds for one of the orders because it was a more like distant family of monocots. And then the second was we were able to get seeds for 2 of the species in this order, but they were not able to like germinate. So we were not able to graft that. So we were quite interested as a lab to understand about photosynthesis, and I'll come to that in a second. So we wanted to see, can we actually graft across this monocotyledonous clade? So can we graft species that are present in this BOP clade, where rice and wheat is present, to this PACMAT clade, where the millets and sorghum is present? So can we actually do interspecies graft? And here from the example, you can see that we grafted pearl millet to wheat, where wheat was used as a rootstock, and then wheat to pearl millet with the success rates that are like shown here. And here you can see the different combinations of grasses that wheat grafted. So for instance, a pearl millet scion, so a pearl millet shoot stalk to a wheat rootstock, and then a rye scion to a sorghum rootstock. So we were quite happy because we were able to not only establish that we can graft monocots, but we could actually show the breadth and extent of grafting to graft it across the whole monocotyledonous clade and also interspecific grafting. So now in the next few minutes, I'll talk about what exactly and in which direction we are taking it forward. So there's a lot more work going on in Cambridge on different aspects of grafting, and Anup is a tutor here, so you're more than welcome to like go and bug him with all the questions. So I'll talk about like some of the aspects that we are testing in the lab at Essex, because I've just started my research group. And what I'm quite passionate about is thinking about not just sustainable agriculture in terms of a dry environment, but also like sustainable use of water. So one thing that we see, and that was quite highlighted in the recent UN summit, was that we are using water unsustainably. And one of the bigger drivers of water is cultivation of these crop staples. And if you think about like agricultural activities alone is accounting for 80% of water usage. And if you think about like rice, which is a crop staple that feeds half of the world's population and usually like responsible for 50 to 80% of the daily global caloric intake, is not water use efficient. And it is a very intense crop that uses water, with almost 45% of total water utilized for global irrigation. So then going back to the monocot grafting thing, can we actually make rice C4? And I'm not making rice C4 because there's been tremendous efforts by brilliant scientists, and they are probably making excellent progress on that front. So what I'm trying to do is, can we actually learn lessons from how C4 has evolved? So C4 photosynthesis is an efficient version of photosynthesis. And can we actually look across this whole angiospermic clade and understand a little bit about the evolution of C4 photosynthesis? And what you can see in these red lines are different families where C4 photosynthesis has evolved. So then going back to that example of grasses, if we think about it, this is the BOP clade where all our global staples are present, such as rye, wheat, and rice. And they perform C3 photosynthesis, the less efficient version of photosynthesis that does not have that higher water use efficiency. But then if you look at the pack-mat clade of grasses, they have all these millets and maize and sorghum that uses C4 photosynthesis, the higher water use efficient grass. And now, because it's very topical, 2023 has actually been called the year of millets. So why not think about how we could integrate and diversify our crop portfolio? Why can't we just not think about wheat, rice, and maize, but also think about millets and sorghum? How can we not be eating sticky rice, but like sticky millet maybe? So thinking about how can we make water smart grafts? With grafting, can we actually have a rice plant with the root of millet and then answer some of the fundamental biology questions? First, is the plant becoming more water use efficient? I have some preliminary data to say it does, but then Russell was like, you can't present graphs. So of course I'm not presenting any graphs here. And then looking at some of the basic biology questions. So what are the signals that are traveling from the root to the shoot or from shoot to the root? And thinking about, are there mobile signals? How the millet root actually affects the photosynthesis of the shoot. So I think it's just a wonderful, wonderful field that we are stepping into. And there are many questions to answer, and we're just at the beginning of it. And with that, I would like to thank the people who were involved in this study and like the collaborators that we are working with to answer and understand monocot grafting a little bit more. Thank you so much for your attention.